Okay, 3.4.1, DNA, genes and chromosomes. So this is part of section 4 and the first video in the series. So what I'm going to do here is just introduce the structure of DNA, genes and chromosomes, and make sure we're comfortable with those terms and what the job of DNA is to do. And then we'll go on and talk about in another video about the ins and outs of protein synthesis and RNA and the part that that plays. So DNA stands for deoxyribonucleic acid, and the DNA is made up of a series of monomers called nucleotides. So this whole thing is a nucleotide. It has a sugar called a deoxyribose sugar, which is where the name comes from. And that deoxyribose sugar is a 5-carbon sugar that is missing an oxygen that differentiates it from ribose sugar. Go back and look at our video on nucleic acids for a bit more detail on that. And then we have a base made of nitrogen, and we have a phosphate. So when this links together, we get a polynucleotide chain. Now, DNA is a double-stranded molecule. So we have two strands that are running in opposite directions. They're anti-parallel. And between the two strands, we have hydrogen bonds that bind in complementary base pairings between A and T bases and between C and G bases. So this binding and this pairing is going to become important in the genetic code later as we see protein synthesis in our next video. So DNA itself, however, has kind of various sections and terminology that we've got to be comfortable with. So what we're looking at there is this twisted double helix, kind of unwound in that picture where I've drawn it in red. This is what it will kind of look like when those bonds form, and it will coil up. Now, it has to be coiled for structure, so to make sure that it's not going to degrade, but also to make sure it's compact to enable lots of genetic code to be stored. We're talking 9 billion letters of code here that has to be compact into the nucleus. So that's a lot of information. So it has to have this structure to enable that. So the DNA itself is broken down into sections. So each sort of section of code, if I put a few more kind of random letters in here, let's say I've got a G here and I've got another T here and an A here. This is one section of DNA along the whole of the genetic code there. And this section, in this part, we're going to call a gene. A gene is just a section of DNA that codes for a particular characteristic, or codes for a particular protein, if we're talking in A-level detail. So the gene provides instructions that ribosomes are going to use in order to form an amino acid sequence, a primary sequence of a protein. And that will be important later. So we get different versions of these genes. These genes in different versions we call alleles. So for instance, you might have a gene that means that you can roll your tongue, whereas another person would have the same gene, but they would have a different version of it, and that would mean they couldn't roll their tongue. Or you might have a gene for attached earlobes or unattached earlobes. We're talking about the same feature, we're just talking about different versions of it, and that's what we mean by an allele. Now, all of these different genes, these 20,000 genes roughly that we have on the DNA, are all coiled up in a structure called a chromosome when the cell begins to divide. But what a chromosome really is really tightly wound DNA that's wrapped around a histone protein. And this is kind of like when you wrap knitting wool around like a ball of yarn or something like that. So it just enables the DNA to be compact and stored really tightly. And when we look at A2, we can talk about acetylation and methylation and how that can inform epigenetics, which is a new study in DNA. So essentially when we're looking at a chromosome, we're looking at a histone DNA complex, and that's important. When we see a chromosome, and we'll get onto this in cell division, what we're looking at is usually kind of sets of chromosomes that look like this in pairs. So we call these pairs homologous pairs, and that's because each location that a gene is found on, which we call a locus. This is the position of one gene. Here's the locus of a second gene. But a homologous pair of chromosomes exists because we have the same genes, just different versions of them, alleles, on either of the chromosomes there. And we'll look more at chromosomes and cell division in a later video. So what we've got there is the homologous pair of chromosomes, one copy that we get from our father, one copy that we get from our mother. If we ever see chromosomes that look like this, what we're looking at there is a replicated chromosome, where during interphase of cell division, one of the arms of the chromosome has been doubled up. All right, so we've talked about how DNA is broken up into sections called genes, and we've talked about how those genes come together to form the whole chromosome, which is a complex of histones and DNA. But there's a few other sort of key terms that we've got to be aware of and key points about the DNA. We say the DNA is a universal code. And what that means is the universal code of DNA is used in all living organisms. So if we looked at a plant, or we looked at bacteria, or we looked at a fungus or animals, all living things use the same genetic instructions, the same DNA. And that's evidence of evolution, that we've all evolved from the same kind of lineage. So when we see that term, that's what this means. We also see the term non-overlapping and degenerate and triplet or codon. Now, this is all kind of key terminology that we've got to have a grasp of, and we'll see will help us explain the later processes. So what we mean by each is non-overlapping essentially means that the code is read in sections of 3 bases called triplets, which we call at A-level codons. You would have encountered triplets at GCSE, hopefully. So sections of 3 bases, as we'll see in the video on protein synthesis, code for one amino acid. And when we put those amino acids together, they form a polypeptide. But this coding, these bases, never overlap. So each triplet codes for one amino acid, each set of 3 bases, and that can't kind of be mixed up. However, these bases, or these codes, can code for for the same amino acid. So let's imagine we've got a triplet here, CAG. That would code for, let's say, and I'm completely making this up off the top of my head, I would have to look it up. Let's say that codes for glycine. CAC might also code for glycine. And what we're saying there is that this base, this final base, the G, has changed, giving us a different base code, but that still codes for the same amino acid. So we say the code there is degenerate. There are 64 possible combinations of triplets. We know that because there are 4 bases and there are 3 positions in each triplet, so 4 times 4 times 4 gives us 64. So there's 64 possible combinations, ACT, TCT, AAA, TTT, yada yada yada. So all of these codes, each code for an individual set of 3 bases, but there are only 20 naturally occurring amino acids. So that therefore means that some of these sets of 3 bases must overlap, like in the example we can see below. So sometimes that causes a bit of confusion about what those terms mean and how that's all linked together. So getting back to this being universal, we have to kind of know that in prokaryotic cells, in bacteria, the DNA isn't as long and it's not associated with histones. We might remember from the stuff on cell biology when we drew the cells out, we've got a cell wall, we've got plasma membrane, we've got cytoplasm. We don't have a nucleus. If we looked really closely at this DNA, it would kind of be short and circular. It would be in a loop. That's because the prokaryotic cells are single-celled organisms. They don't require as many proteins as multicellular organisms. So eukaryotic cells generally have longer DNA as a result of that. Remember, bacterial cells also have these small extra copies of DNA called plasmids, which can be used for transfer, and they're not to be confused with that. So that's kind of pretty much it in terms of understanding all the key parts before we look at DNA and protein synthesis. The final part we need to know about is that when we look at DNA, if we looked at this as blocks of genes, blocks of information, So let's say we've got lots of sections of DNA here. There'll be sections within each gene that don't actually code for anything that's going to be used in the protein, i.e. their bases give us sections of DNA that aren't going to be translated into a protein by the ribosome. So what happens is they're removed by a protein complex called a spliceosome. And this spliceosome carries out the process splicing to remove those sections of non-coding DNA, which we call introns. So what's left at the end of it, once all those introns have been removed, i.e. all the bits that aren't green there, all of that DNA are classed as exons. So those exons that are left behind. Right, we're going to stop this video here, Mr. Editor, and stick this second bit on the end of that.